Speaker. Can everyone hear me fine? All right. Dr. Bix is a professor of neurosurgery and neurology at Tulane University, and he's also currently the director of Clinical Neuroscience Research Center, vice chair of Clinical and Translational Research in the Department of Neurosurgery, and the Vada Odom Reynolds Chair in Stroke Research at Tulane University. He holds the position of clinical lecturer at the University of Glasgow, adjunct professor at the Queensland University of Technology, and honorary professor at the University of Manchester, and he is a fellow of the American Heart Association. He completed his clinical and research training at Baylor, University of Pennsylvania, and Thomas Jefferson University. He has published more than 50 papers in reputed journals and has won several research awards. He has several patents for his various scientific discoveries, and is currently the PI of multiple NIH-funded trials. His research focus is the role and therapeutic potential of the extracellular matrix and its receptors in stroke and vascular dementia. Please join me in welcoming Dr. Gregory Bix for this talk.
Thank you. Pleasure. All right, can everyone hear me okay? Hopefully online. All right. Well, I want to say thank you so much to Dr. Deb Gustafson and colleagues for having me here. That week between, I guess, Christmas and New Year's can be a pretty quiet week, so I was thrilled to have the opportunity to travel up from New Orleans to New York. All right, is that, let's see. Okay, we're good. All right. So today I am going to sort of give you the greatest hits of my research program over the last, well, since 2007. I'm going to start talking about ischemic stroke. Then I'll talk a little bit about vascular dementia, and then various therapeutic pathophysiologic targets that we've identified. And then I'll end on some of our work on COVID and the neurologic manifestations of COVID and long COVID, which relate very much to the earlier projects that I just mentioned. So I don't think I need to tell a group of neurologists that stroke is bad and that it's pretty prevalent. And I think you all are pretty familiar with that. This pointer, no. If I move this, can you guys see it on the screen? Okay, perfect. So this is where I practice right now. In terms of mortality, it's not so great in the South. And it's interesting that, you know, even when you factor in things like diet and physical activity and weight, it still doesn't quite account for the high morbidity and mortality rates of stroke in the so-called Bible Belt. A lot of it, I actually think, has to do with how close you are to a primary stroke referral center or care center. Anyway, my home state doesn't do particularly good in that. We're number 3, the number of deaths for stroke. And I'm going to be talking about ischemic stroke specifically. So I think an important concept that is often overlooked is that stroke affects more than just neurons, right? And that a number of years ago, a friend and colleague of mine, Mike Moskowitz in Boston, coined the term neurovascular unit. And this refers to not just the neurons, but the blood supply and the supporting cells that are structurally and functionally integrated with neuronal health and viability. And that includes things like, of course, the blood vessels and endothelial cells that make them up, the basement membrane, which I'll talk a fair bit about, pericytes that support, astrocytes that sort of link between blood vessels and other neurons, et cetera. And this sort of interplay in the blood-brain barrier all is impacted directly and indirectly by stroke. I spend a lot of time thinking about the vascular basement membrane, which is sort of that zoomed in. And I forgot to mention, many illustrations sort of show the matrix as this amorphous space. You can see it here. It's this yellow blob. But I spend a lot of time thinking about the matrix in the context of how it's in the right place at the right time to directly and indirectly impact the neurovascular unit. In particular, I have a favorite matrix component, something called perlecan. Perlecan was coined because on electron microscopy, it looked like a bunch of pearls on a string. And it's something called a heparan sulfate proteoglycan, which essentially means it's a protein with GAG chains. It has 5 domains, which are very cleverly named 1, 2, 3, 4, and 5. The 5th domain has a proteolytic hotspot that can cleave it from the other domains. And there are a number of proteases that have been identified over the years that can do this. But upon cleavage, this fifth domain, this protein core, can interact with cell surface receptors and affect complex biologic systems. So Perlecan itself, each one of those domains has proteins with structural homology to other proteins. And so it's sort of been described as a sort of scaffold that could bind and interact with other proteins. And then when things are cleaved, have different activities than the whole. So the parts are actually different once they're liberated from the protein core. What do we know about Perlecan? We know that it's pretty much found wherever there's a vascular basement membrane. So wherever a blood vessel is, it's got a basement membrane, there's Perlecan. It's also prominent in neuromuscular junction, where it helps sequester acetylcholinesterase. So there are lots and lots of different and interesting issues when Perlecan is disrupted. And another clue that it's important is that it's highly evolutionarily conserved. So from C. elegans and Drosophila all the way to man, it's very, very similar. There's differences in domain 4 in terms of the number of immunoglobulin-like repeats, those little yellow balls right there. But we know, and this was a relatively earlier observation, that if you're physically active for a period, say you go jog a few miles, and then you give a urine sample, and they do a proteomic analysis on your urine, typically there's not supposed to be a lot of protein in your urine, right, unless there's kidney damage. But to a person, when someone is active, one of the only proteins that's found with some specificity is a piece of domain 5 called LG3, which is the C-terminal most portion. So that was a clue that Perlecan plays some sort of a role in the vascular tone and vascular regulation, and perhaps links to the relevance of exercise to vascular health. As I sort of alluded to, life without Perlecan has its problems. It's highly evolutionarily conserved, as I indicated. So in addition to in vascular basement membranes, it's got a prominent role in heart development. It is also found in cartilage. So a lot of the very rare autosomal recessive human conditions that are linked to Perlecan involve musculoskeletal phenotypes, myotonia, chondrodysplasia, that sort of thing. In mice, it's been challenging to study it in translational models because, as you might guess, if you get rid of it altogether, it's embryonic lethal. So we've had different tricks, and I'll talk about some of those. All right. So I'm going to cut right to the chase because I am a translational and clinical neurologist. I spent 5 years as a postdoc in Philadelphia studying this molecule, this domain 5, as a potential therapy for cancer. In 1997, a guy by the name of Judah Folkman came out with a Cell paper that suggested that the extracellular matrix upon proteolysis could liberate little protein fragments that could block solid tumor growth by blocking new blood vessel growth. And solid tumors need a blood supply to grow and then to hematogenously spread. And so we looked at our protein fragment, domain 5, and saw that it seemed to suppress blood vessel growth in tumors. Then I started my own lab in 2007. I'm at Texas A&M. I moved from here, or this area, to the south. And I had done my MD and my PhD work in neurology and in brain development. And I thought, what am I going to study now? I've spent 5 years doing cancer work. Then I realized that a lot of brain injury essentially is a recapitulation of developmental pathways in response to injury. And so I've spent my PhD studying brain development and lissencephaly and neuronal migration, and then my postdoc studying blood vessel growth. And I looked to see what was known in the literature about angiogenesis, so-called new blood vessel growth after brain injury and stroke. Turns out not a lot. And I thought, okay, well, maybe Perlecan plays a role. And I couldn't find all that much relating to Perlecan in the brain other than that it was expressed. And I thought this is an interesting area that I could actually explore and leverage what I already understood and what I could learn, hopefully into a new therapeutic area. And I guess I'm talking to you about it many years later, so it did work out. And so the questions that I asked when I started my lab, essentially, domain 5, is it ever generated in the brain? So it's one thing to say that there's Perlecan present coating blood vessels in the brain. It's another to say it's actively being processed into domain 5. And then I was interested in stroke. Does stroke influence degeneration? And then does it mean anything? So it's one thing to say that it's there. It's another to say that it's processed, but is it clinically or therapeutically relevant? And yes, can we exploit that therapeutically? And so in 2007, literally the year I started my lab, a paper came out that said, yes, if we take normal human cerebrospinal fluid and do proteomic analysis on it, Perlecan LG3, which is that on that 2D STS page shell, the red, that's LG3. But I've learned from years of experience that you can't trust everything you read. So the first thing I did was get some human CSF from patients that had pseudotumor cerebri, and they were getting therapeutic LPs. And we probed for Perlecan, and we were able to identify in a number of patients that same 25, 26 kilodalton LG3 fragment in domain 5 was about 80, 85, and that there. So that was a plot that I ran. So relevance to human stroke. So everything that I'm talking about, regardless of whether it's studied in mice or rats, I start in human and kind of work back into the lab and then go back to the clinics. So what you're looking at here is brain tissue from patients that died, unfortunately, from ischemic stroke. We had to go all the way to Japan because they had the biobank for that at the time. And what we noted was interesting. So this is penumbra or peri-infarct areas in patients that died at different points. A control was just someone who just passed away. And the brown is stains for Perlecan. And so what we noticed, typically you would expect to see what you see in the control. It's staining blood vessels because it's vascular basement membrane. A couple of days after stroke, patients that died, all the Perlecan's gone. We saw next to no immunoreactivity for Perlecan. So that's pretty abnormal. 7 days later, a little bit has come back, but not a lot. But then interestingly, 3 months, and we've now validated this 6 months, a year, a few years, there's a lot more of it than you would expect. So there's something happening with Perlecan. It goes from being present to being absent to being overexpressed. And so that sort of gets to the, is it there? Is it processed, clinically relevant? This is people. So we did a couple of things. So remember I mentioned that Perlecan, embryonic lethal, if you get rid of it, you don't get viable embryos. Interestingly, the embryos, I think it's day 15 or day 16 in utero, they have 2 types of abnormalities, cardiac and brain. And so a lot of work had been done looking at the effects of Perlecan deficiency in the heart, but I was the first to actually look at it in the brain. So the clues were always there. And these papers came out in the early 2000s, but I think I was the first person to seriously explore it in the brain. Anyway, if you take mice and knock down the Perlecan, but not completely, so you get rid of about 90% of it, you get viable, fertile mice. When you stress them, there are clear differences. They have some interesting phenotypes. They've got arthritis and some other issues related to the musculoskeletal phenotypes I alluded to earlier. But if you stroke them, a couple of things. One, if you stroke a normal mouse and then look, you see domain 5 being generated. That's the Western blot in panel A. But if you take a Perlecan hypomorph, I'm just going to call it Perlecan knockout, there's much less domain 5 generated because there's much, much less Perlecan, about 90% less. But then when you look at the brains of these mice, after you've experimentally stroked them by pinching a middle cerebral artery, every day you look at the infarct, it's larger. So there's a functional consequence or clinical consequence of Perlecan deficiency, which is bigger strokes. So that was kind of an important thing for us. And so getting to the actual therapeutic angle, here's what I posited back in, I think, 2008. Domain 5 is important. It actually is part of the brain's innate response to injury. It's generated after stroke. It does things that are potentially beneficial. When you get rid of it, or there's much less of it, things are worse. So maybe we can administer it after stroke and see a better outcome. And there are different things one might look at after an experimental stroke. One is, does it keep cells from dying, so-called neuroprotection, focused specifically on neurons here. And then the other is, does it foster repair? So neuronal or brain repair is something that we understand even less about than protection. But there are different aspects of repair that I focused on in my work. One is blood vessel growth, new blood vessel growth, so-called angiogenesis. Another is neurogenesis, new neurons, and then astrogliosis. And I won't have time to talk about all of that. I'm going to go right to the therapy. So one of the first things we did, whenever you're testing something that you think has an effect on the brain, but you're giving it in a vein or IP, is does your therapy actually go where you want it to go? Or do I have to drill a hole in the head and inject it right into the brain? So this is a rat brain section. It's stained for DAPI, which is a nuclear stain. And the pink is a tag that we attached to the protein that we injected in the abdomen intraperitoneally. And this is an animal that had been stroked. So what you're seeing, this area right here is the ischemic core, and then this is the penumbra. So by and large, this is about, I think 8 hours after we injected, we induced the stroke. We injected right after the perlecan domain 5. This is human recombinant protein, I should point out. And then 8 hours later, we sacked the animal and we stained, re-imaged. And you can see that it seems to track very nicely to the site of injury. We didn't see very much of it in other parts of the body over time. There's many reasons for that. I'll talk about some of them. A bit of a complex slide, but suffice it to say, animals' strokes evolved in size over time. So these are different cohorts. This wasn't serial imaging, just animals where we stroked them. These are now mice. We looked at infarct volume on day 1, day 2, day 3, 7 days out, and then 15 days out. And we started treatment with the protein pretty soon after stroke. We've since extended the therapeutic window. But one of the interesting things that we saw in sort of a dose-dependent fashion with the protein was that we could arrest the expansion of the injury. So if we treated early and we looked early, we didn't see anything much. But if we treated and then looked out later, stroke never got bigger. And I actually think this was 24 hours. Yeah. So we treated on day 1. And then you can see that the domain 5 actually prevented the stroke from getting any bigger. We actually did studies with the perlecan knockout mice where we so-called gave rescue therapy, the domain 5, and we could arrest the expansion of the injury in those mice as well. This is just one of the many sort of things we do. You can't ask a mouse to do things on demand. So you put it in situations where it doesn't want to be, and then you can look and see whether it uses the stroke affected paw or side of the body. And this is a cylinder test. This was a rat study that we did. And essentially, if they've got it, well, I don't have to explain this to you guys. Essentially, animals after stroke don't use the stroke affected side. They're the contralateral paw. With treatment, we saw a return to a 50-50 baseline within about 24 hours, which persisted over a couple of weeks. So this is, first major study that came out of my lab as an independent PI back in 2011. More recently, we started giving the domain 5 a week after stroke. And for those of you that treat stroke, you know that there's not a lot of effective therapeutics that can be initiated chronically that do much. I know I'm familiar with some work. Some physical therapy is helpful. Antidepressants, when given chronically, can actually help with functional recovery, but there's not a lot. So we're particularly excited about this study because a week after we stroked the animals, we gave them some of the protein, and we saw improvement on rotarod study as well as grip strength in rodents. And this is, you can see many years later, we're still at it. So I'm excited about this work because it's about to enter phase 1B clinical trials with a company that licensed my patents. I learned all about patents and how you can't disclose your ideas and then try to get a patent, all this stuff early in my career. But essentially, domain 5 is an endogenous protein fragment that has clinical relevance that in experimental models of ischemic stroke, we could administer and see therapeutic effect. And I haven't had a chance to really talk about all the different mechanisms, but essentially we see that neurons love it, so it's neuroprotective. We see that it's stimulating.
So call the doctor's office and let them know that you really think he has a fever. If it would make any difference to their diagnostic.
It does make it.
I think
It does?
It does.
Oh.
I don't know how to mute anyone, so I'll just keep going. We did a lot of work to show that it stimulated new blood vessel growth after injury, which is important to actually support neurogenesis, which is important understanding almost like the tissue regeneration. And we also looked at scarring. Scarring is a bit of a yin and a yang sort of thing in the brain. It was primarily thought to be bad, but we now know that it has also some significant benefit, and domain 5 also modulates that. So I need to update this slide because the clinical trials are starting next month. I'm sorry, I can't hear you. Yes, that was the very first thing we had to do, for the FDA. And what's interesting is one of the things that TPA does, I don't know if it was on that slide, is it actually cleaves perlecan right to solubilize. So we're seeing enhanced benefit when we give domain 5 with TPA, because we think there's an endogenous cleavage of perlecan as well as the domain 5 administered. So it's a very good question. So yeah, I sort of alluded to this, but we spent about 5 or 6 years really looking at mechanisms of action. And if this is a clinical audience, I don't want to spend a lot of time going over kinases and phosphorylation and that sort of thing. Suffice it to say, we identified a family of receptors that I'll be talking about more or less for the rest of my talk, called integrins. And I had previously identified a alpha-2 beta-1 collagen receptor for my work in cancer. But it turns out that in the brain, endothelial cells that make up the blood vessels don't express that receptor. They express something called alpha-5 beta-1, which has a high affinity for perlecan domain 5. We identified a lot of VEGF, vascular endothelial growth factor, primordial first identified growth factor that impacts blood vessels. And that seems to have explain a lot of the effects we're seeing with domain 5. Not all, but a lot of them. All right, so integrins, and why should we care about these receptors? Integrins are found in every cell in the body, and depending upon what complement of integrins a cell will express determines what it can stick to. So I'm interested in primarily endothelial cells in the brain, and they express a handful of different integrins, and this is very context dependent. In other words, an endothelial cell in the brain will express one family of integrins during brain development, a different in a mature brain, and then there's a reactivation of developmental receptors upon injury. And that's pretty much all you need to know about that, other than they're heterodimeric, which means an alpha receptor or subunit is paired with the beta, and depending upon which alpha associates with which beta, you get different affinities for different matrix components. So I'm particularly, I already mentioned, interested in something called alpha-5 beta-1, which binds to fibronectin. Fibronectin is a provisional matrix, so it's found in the brain during development. There's almost none of it in a mature brain. Then upon injury of any kind, whether it's a vascular insult, stroke, vascular dementia, Alzheimer's, MS, ALS, pretty much any neurodegenerative disease you can think of, alpha-5 beta-1 is upregulated in the blood vessels that are impacted. So this receptor signals from inside the cell out and outside the cell in. And this family of receptors are the primary receptors that interface with matrix, whether it's extracellular matrix or vascular basement membrane. And they play a profound role in regulating blood-brain barrier integrity, which is highly relevant to everything I've spoken about and everything I'm about to talk about. So some work back in the day linked beta-1 integrins in general to impacting something called tight junction proteins. Tight junction proteins are sort of what help stick the endothelial cells together. And when various integrins are activated or blocked, you can either tighten or loosen those connections. So we and others have done a lot of work showing that alpha-5 beta-1, as I already sort of alluded to, goes up. Its expression goes up, within hours after an ischemic stroke. And it doesn't matter whether it's in a person, we've validated it clinically, but in our various stroke models, there are really two fundamental ways to model stroke in a rodent. You can transiently occlude an artery or permanently. And what we know is that alpha-5 integrin goes up in the core and the penumbral region within 24 hours after. This is just one of many examples. You're looking, the hashtag indicates the ischemic core. This is immunofluorescence for alpha-5. It just goes up exponentially between days really 2 and 4. But also note that it goes up right around the site of injury. It's not a pan-brain upregulation. It's really ipsilateral to the infarct. All right, so when we're studying these things like the protein, like perlecan in mice, we try to knock it out, we try to upregulate it and see what happens. Richard Hines at MIT, who won the Lasker Prize for this work, actually, which is right before the Nobel Prize, developed various ways to knock this receptor out. Of course, it's important, so if you get rid of it altogether, it's embryonic lethal. So he knocks out in endothelial cells. And interestingly, I think disappointingly to him, they weren't all that abnormal. And that's because there are other integrins that can sort of cover in the absence, other receptors that can bind the same ligands that can functionally compensate. But we got our hands on the mouse. He shipped them to us because we were interested in initially looking at the consequence of this receptor being absent for our perlecan domain 5 therapy. Remember, I mentioned that this receptor was important for that therapeutic. So the hypothesis was if there's no alpha-5 beta-1, this perlecan domain 5 will lose its therapeutic effect. That was the idea. But the reason we do research is you never know what the outcome is going to be. And I can tell you that at the time, a friend and colleague of mine, Richard Milner, out in San Diego, didn't know how to do stroke. So he did the next best thing at the time, which was put them in a hypoxia chamber. So he took these mice and basically put them on Mount Everest for a period of time, in this case, I think 2 weeks. And when you do this, the brain doesn't like to be chronically hypoxic. It'll eventually start to grow new blood vessels to try to compensate. And it was interesting because when he looked, he saw that normal mice, within a few days, 3-4 days, will start to grow new blood vessels, but the alpha-5 beta-1 endothelial cell selective knockout mice don't, at least initially. But eventually, you wait long enough, they do. And so the conclusion, I always rip him about this because his conclusion is wrong. At the time, he said, well, they're impaired in angiogenesis. And I should have mentioned this earlier. This receptor is critical for new blood vessel growth, alpha-5 beta-1, upregulated after injury. It's a provisional integrin, grows new blood vessels. Anywhere in the body, alpha-5 beta-1 is upregulated, you get new blood vessels sprouting. So my thought was simple. Maybe it's not that these mice were impaired in their ability to grow new blood vessels. Maybe they were resistant to the hypoxia to begin with, and it took almost twice as long for them to start to grow new blood vessels. And I think we now, Richard and I now agree that this is correct. So I mentioned that we started to stroke these mice, and the idea was simple. Let's stroke them, and then we'll go back and treat them with domain 5, and we'll see the domain 5 has lost a lot of its punch, and that'll be the nice AIM3 and the RO1. But the surprise was that when we stroked them, we didn't see much injury. This was a transient stroke model, and I can tell you that we've more recently confirmed it with a permanent model where you permanently occlude the artery. We did something called TTC stains, where you take the brains, you stroke, you wait different periods of time, you get the brains, you stain them with this TTC, viable tissue. The mitochondria turn the white TTC red. If the area is metabolically compromised or dead, it's white. So when you see those sections of brain, the white that's infarct. And lo and behold, the mice had little to no infarct, the alpha-5 knockouts. This was done right before I moved my lab from A&M to Kentucky, at the University of Kentucky. And so I repeated, the first thing I did was, well, maybe there's something different about the water in Kentucky, limestone and all that, who knows? So let's repeat it. We did, got the same result. I thought, that's interesting. There's nothing to treat. So we couldn't pursue the domain 5 story, but I realized pretty quickly that this was maybe a complete new therapeutic angle. And this is, more of the same. We stained for cell death. We didn't see much of any. And these alpha-5 knockout mice 2-3 days later, this is just an apoptosis tunnel stain. This is recent. This is some data that we did about a month ago. where we took the same alpha-5 endothelial cell knockout mice, and we did a photothrombotic stroke. Essentially, you inject a dye that's sensitive to light, and when you shine a laser point source on it, it causes coagulation. You basically get blood clots, and you get a permanent stroke. People sort of liken it to a TBI more than a stroke, but it's very pinpoint in accuracy. So 100% of the time, you can get a stroke, an infarct, wherever you want it to be, and however large you want it to be, which is kind of nice. And when you're modeling brain injury, to cause an injury that's 100% of the time going to be the same. Anyway, we did that in these mice, and we carried the experiment out, and this study's still ongoing. You're looking 2-3 weeks out, and look at this is on a rotorod. It's just a turning thing that the rodents are running on. And if they're neurologically functionally impaired, they fall off pretty quickly, right? And there's a bit of a learning component as well. But look at the performance in the stroked knockouts compared to the wild type. They separate pretty early on, and they continue to get better. And they've gone from baseline, they're above it. So they essentially, there's no negative effect. Again, permanent stroke model. All right. So why do I think this is happening? And this is going to be very relevant to how I'm going to end the talk talking about COVID. We think that this has everything to do with blood-brain barrier. So earlier I mentioned that this receptor is critical for new blood vessel growth. And one of the first stages in angiogenesis, which I should mention, is blood vessel sprouting from pre-existing vasculature. That's angiogenesis. Vasculogenesis, by contrast, is de novo blood vessel growth. It's an important distinction because about 95% of all the blood vessels in the brain are angiogenic in origin. You get the initial large arteries, the ACA, MCA, PCA, and then everything sprouts off of those. So when the brain is injured, it's an angiogenic response that occurs 3 to 7 days after the injury. And one of the first stages of that are the endothelial cells that are present have to pull apart because they ultimately will start to move and change their shape to make a new blood vessel. And the receptor that's critical for that is alpha-5 beta-1. But a negative consequence of cells that are making a barrier pulling apart is the barrier opens up. That's probably not a good thing right after a stroke, right, or other brain injury. So we think that blocking this receptor being absent, has everything to do with the barrier being disrupted early on. And a consequence of disrupting it or blocking it is maybe there's less of that. So the barrier is more resilient. There's less swelling, less inflammation. So all those cells that like to infiltrate the brain, neutrophils, macrophages, et cetera, there's less of them. And so less primary and secondary injury after stroke. And I'm not going to walk you through this. Suffice it to say that the blood-brain barrier opens up repeatedly after stroke. And I don't need to tell a room full of neurologists that, it's that tertiary opening, a couple of days out where there's a lot of swelling and inflammation, edema, et cetera, that we think is the most therapeutically worrisome. So we did a deep dive looking at blood-brain barrier disruption after stroke in these mice, and we couldn't really find any. So this is just staining for IgG. If you've got megadalton size IgG in your brain parenchyma, that means the barrier is Swiss cheese. It's just full of holes. You should never have IgG in the brain. So yeah, normally we see lots of it after stroke in mice. In the knockout mice, we don't see much, if any. All right. So obviously, we're not all walking around deficient in alpha-5 integrin. Remember, it's embryonic lethal, so none of us are suffering from that. But there are therapies that can target this receptor. And for me, it was sort of full circle. I spent 5 years doing cancer research. And I'll tell you that the people that study the heart and study blood vessels and tumors are about a decade ahead of those of us that study the brain and cerebrovascular. Most of the tools that I've ever used in my line of work have come from cancer or cardiology. And one of them is something called ATN 161. And the more I study this, the more wowed I am by what this peptide can do. So this is a 5 amino acid peptide that was developed from a synergy domain in fibronectin. It can bind to all fibronectin integrins but it doesn't do it randomly. It prefers integrins that have been primed for activity. And that's important because if you give antibodies to integrins, they bind to the integrin wherever it's located, regardless of whether it's being primed to do anything. And that matters because if you give an integrin-based therapy, it can go all kinds of places and have undesired side effects. ATN preferentially binds to and antagonizes activated integrin, activated alpha-5 integrin specifically, and that's upregulated in the brain after injury. Remember I showed before. And it's been clinically validated. So again, everything I talked about started in people. This is a peptide that was given to patients with glioblastoma and metastatic melanoma. It was given 3 times a week IV infusion. It actually stabilized disease. The problem was it didn't add appreciably to patients' lifespan. And so the company sort of went belly up, but not before their patents expired. And their chief science officer basically gave me a freezer full of the peptide and about a 2 terabyte data dump of all their stuff. So I've been using it ever since. We have a number of provisional patents, or rather utility patents for ATN in various neurologic conditions. So maybe some of you will be using it in the future in the clinics. But I'm using it now for stroke, for vascular dementia, and we've published in COVID. So I'm just going to go through this quickly in the interest of time. Everything that we did with domain 5, we repeated with ATN, and we can give it IV, IA, or IP. And this study came out a few years ago, where we did the same exact study, transient middle cerebral artery occlusion. We could reduce infarct volume with repeated administrations of this peptide, and we could improve functional outcomes on neuroscore, et cetera. And then we had a human neuroradiologist read the MRI scans, blinded, to the treatment conditions, and those were the outcomes. We saw less edema and smaller infarcts in scanned mice, stroke. And this is just a stain for IgG, same thing that I showed before. We see much less IgG extravasation into the core and the penumbra region in mice treated with ATN after stroke. So I'm excited about this work because this is a validated therapeutic target in stroke that we are advancing forward as well. But there are other indications that I want to spend the remaining time talking about. So again, I think I can skip this. You all know vascular dementia is bad, and probably you treat a lot of patients that have mixed dementias with Alzheimer's and vascular components, and there's a lot of controversy whether they're really separate etiologies. Symptomatically, they present differently, but I've yet to see an Alzheimer's autopsy brain specimen that doesn't have mixed vascular components. How do we model this in the lab? Mice on their own don't get demented, so you've got to kind of coax them along. And there's 2 ways to do it. You can modify genes, either add human genes that induce dementia like ApoE4 with a bad diet will give mice Alzheimer's and demented phenotypes. But one of the ways that we do it is we put coils on the carotid arteries bilaterally. It's called bilateral carotid artery stenosis. And you can reduce but not eliminate blood flow to the brain, causes white matter injury over a couple of weeks. And by about a month, these animals have cognitive deficit on a battery of cognitive tests. If you leave the coils on long enough, eventually gray matter, hippocampal injury, et cetera, will ensue. But I like it because you can take these coils and put them on mice of any stripe. So if there's a gene you're interested in, you think it may have some relevance to dementia, you can look at it and put coils on there and accelerate things, et cetera. So it's a surgical experimental intervention to induce a cognitive phenotype in mice or rats. So we did a lot of work characterizing the model. My friend Masafumi Ihara in Japan, it's interesting, there are a lot of, anecdotally, a lot of dementia researchers, vascular dementia, come from Japan. And that's because they have a hyper-aged population. And so they live long enough to have these vascular dementias. They're in their 90s and 100s even. And so a lot of the really good vascular dementia work comes out of that country. Anyway, we borrowed the model from him, and we looked at different things that happen in the brains of these mice early on. And we noted that days into putting the coils on the rodents, the blood-brain barrier starts to break down. And we see it pretty much everywhere we look. And by a couple of weeks out, there's a lot of it. And so the idea is very similar. Could we mitigate the effects on cognition in this model by blocking alpha-5? And I'm going to kind of buzz through this in the interest of time, but treating with ATN in this model, we could physiologically and pathophysiologically disrupt a lot of the brain changes. So alpha-5 goes up in this model. We could prevent that by blocking alpha-5. We looked at astrocytes. So GFAP is a marker for astrocytes. We also looked at vimentin, but we could block astrocytosis in this model. Then functionally, we used here a Y maze, but we also did Morse water maze and other cognitive testing. And we saw a nice impact with ATN. Skip that. I want to get to this. Every time I talk about COVID, I try to put in a cool image. And this was I think the happiest one that I could find. It looks kind of like a piñata, at least to me. But I don't think I need to tell this audience all that much about it, other than we've all sort of lived through it the last couple of years. But an interesting observation was made in March 2020. And I want to take you back. As you may recall, that's about when things went pretty south, right? We were on lockdown. And Tulane, I didn't know at the time, has a robust BSL-3 virology infectious disease program. And so we never closed. So my lab was open throughout lockdown. But I thought, if we're going to stay open, I'm not just going to stroke more mice. I'd like to do something relevant to what's happening in the world at this moment. And an interesting article came out from I think this group, I think in Norway or Sweden. Anyway, they noted that there was something special about SARS-CoV-2. And what you're looking at is a map of all known coronaviruses at the time and the amino acid sequence of spike, and specifically the part of the spike that binds to ACE2. And what they noted was that of all the various coronaviruses, SARS-CoV-2 had something unique. And that's highlighted here. This 3 amino acid sequence, RGD. I didn't talk about this before, but RGD is an integrin binding motif. Alpha-5, beta-1, and about 5 or 6 other integrins all are in the RGD family. So I thought, that's interesting. And in this review article, they speculated might the presence of integrins play a key role in why this virus is so pathogenic, why once you're exposed to it, it gets into cells. Now remember, it had been identified that ACE2 was a primary receptor and is a primary receptor. But so ACE2 was also a receptor for every other coronavirus you see on that slide, but none of them really made us as sick, even the SARS Classic, 20 years ago, right? So since then, we learned that not only is that RGD present, but it's if you look at the UK variant on the slide, it's right where you would want it to be if you were engineering a spike to grab onto integrins. And every single mutation in every variant of concern has done one of two things. Has either modified the protein around it to make that RGD more accessible for binding, or has modified the RGD sequence itself. There's an RGC or RGD, other amino acid that can bind to integrins even better. And there's a lot of controversy in the literature. If you look it up right now, you'll see papers that say, oh, it has no bearing whatsoever on the pathology. I'm in the other camp that says that it does. But regardless, we and others posited that these integrins, which are known to associate with ACE2, in fact, they regulate the expression of ACE2 in many cell types, play a key role in how the virus grabs onto cells and gets in. So early work that we did in mid-March during lockdown showed that with first we did it with spike protein, but then we did it with live virus. We could actually inhibit binding of the spike protein to cells and to integrins with ATN. So we were the first lab to demonstrate that spike could bind to integrin. We demonstrated that my favorite peptide ATN could mitigate that interaction. And I'm not going to walk you through all the variants. Suffice it to say, every variant we tested showed it bound to integrin, and some much better than others, as you might imagine. The viruses evolved to grab onto cells better, not worse. We could inhibit with ATN. But most importantly, VeroE6 cells. These are monkey kidney cells, but those of you that study virus know, excuse me, it's a filet mignon to COVID. So people that culture COVID in the lab grow it up in VeroE6 cells. So if you can demonstrate your drug impacts their viral replication in this cell, that means it's potent, because this is a favorable environment for the virus to turn over. And we were able to inhibit viral load in these cells with ATN in a somewhat dose-dependent fashion. That's just showing you the actual cells. You don't need to be a virologist to know that those hypodense round cells are not happy. And there's a lot more of them when you dump virus on, and there's a lot less of them when you add virus plus ATN. So, excuse me, drink some water. I get so excited. My throat dries. Yeah. Mm-hmm. Yep. Yep. So we think that COVID represents a hypercoagulable state, and not only strokes, but also these so-called microclots that might contribute to long COVID. There's some speculation on that. We think it's activating platelet integrins. And I talk about this, but integrins are robustly found on platelets, especially the collagen ones, and they contribute to platelet aggregation. So alpha 2 beta 1, G2, I'm going to forget the nomenclature, but type 1 collagen is a primary aggregant for platelets. And we noted that as well. 20 and 30-year-olds, otherwise in good health, having these massive MCA strokes, right? And they were so bad, a neurosurgeon of mine who does thrombectomy was telling me that as he would pull the clot out, it would be forming in real time. He called it a clown scarf, because he would keep pulling and it would keep forming. And that was kind of scary to hear. But those particular variants were ones that actually the RGD was more prominent. So I think there's an association there, but thank you for asking. So anyway, this was our early work showing that this peptide had some efficacy. We expanded it in vivo, where we were sort of limited to the models we had at the time. So 2020, 2021, there was no good rodent model. You may remember people were using ferrets and non-human primate, which we actually did as well, but that's slow research. So the very first thing we could get our hands on was a mouse that we could make express human ACE2 by using an adeno-associated virus. Literally, you can put this adeno-associated ACE2 virus in the lungs, and so you could actually get normal wild-type coronavirus, SARS-CoV-2, to infect a normal mouse once it was expressing human ACE2 in its airways. It's good and bad. It's good because you got infection. It's bad because you could not get the virus to replicate outside of the lungs, because ACE2 outside. But it was pretty good to look at infection, yes or no. And when we used this model, we could see that ATN could actually inhibit viral replication in the lungs of these mice that were made to express ACE2 just in the lungs. We then moved on to a mouse that was genetically engineered to express ACE2, but I feel like it's sort of like Goldilocks, right? Where it was too cold and then too hot, just right. So the first mouse had limited ACE2. This mouse has too much. So the consequence of this keratin promoter is that you get human ACE2 everywhere, places that it should be, places that it shouldn't be. So it's not really good for looking at the pathology or pathophysiology of the virus, but it's a better model than the too little. We saw inhibition of viral replication in these mice as well. Again, this was a newer model at the time, but not ideal. Then a friend and colleague of mine, Ralph Barrick at UNC, took coronavirus and passaged it 10 times in the lungs of wild-type mice. And after 10 passages, developed a coronavirus that could infect mice. Now, Rand Paul and people in Congress aren't so thrilled about gain-of-function kind of research. So there's that. But this is literally what it is. You took a strain of a virus and made it infectious in an organism, in this case a mouse, that it normally wouldn't otherwise infect. But for a research perspective, it's wonderful because now we can take any mouse you can imagine and not do anything to it. We can infect it with this MA10, it gets sick, and the illness and the pathology mimics what's seen in people. So you don't have to do anything special other than, you know, BSL-3 and hope it doesn't get out, right? So we in collaboration with Ralph and others did this. And I can tell you, very first thing we looked is make sure it's got that RGD. It does, because, you know, the virus is clever and it can mutate in ways that might make your experimental model irrelevant or not translatable. All right, skip that. So, but now we're looking in the brain. So everything I've told you about was, you know, infection, yes or no. This is brain stuff. And everything in the next few minutes I'll talk about is brain. So if we load these mice up with MA10, these are wild type normal mice you'll find in any lab that does research in rodents, we could impact tight junction proteins. And we could, here you're just looking at Claudin-5. This is just PCR. So a few days into COVID infection, tight junction protein transcription and translation goes down. We also used immunocompromised mice, which are also impacted. So it wasn't an immune phenomenon. Astrocytes, GFAP markers in the brain go up by a lot. This is a few days after infection. We looked at microglial markers. The immunofluorescence isn't great here. It's since been repeated. That goes up. So there is acute neuroinflammation, COVID infection. Again, this wasn't enough virus to, you know, kill the mice, but it was enough to make them sick. They lost weight. And this is just more of the same. Other markers of inflammation in the brain go up. And I'll skip all that. So we did a study where we treated with ATN, that same model. We also used selengitide, which blocks alpha V beta 3. It's another fibronectin receptor that a few people had thought might have some therapeutic impact. The good news is we saw changes, again, in astrocytes that ATN could mitigate. It's just on the cusp of significance there. And I don't want to bore you with all these changes. But there are a lot of things that MA10, so coronavirus does to genes and things in the brain acutely, and we now know chronically that ATN treatment can modify. And serotonin is one that you may have heard about, seems to play a role in long COVID. We saw changes with ATN. Still don't know what this means because I barely understand the science that goes into figuring out these pathways from all the genes that you get from RNA-seq analysis. I'm learning more and more about these new approaches. But long COVID, I'm going to say a couple of things. One, I think that COVID is primarily a vascular condition at this stage. And two, long COVID is the disease. Every time you get a coronavirus infection, that's what you're playing Russian roulette with. It's not crystal clear whether or not repeat infections increase your chance, but I don't think it decreases your likelihood of getting long COVID. And this is what scares me, and this is why I wore a mask when I flew up here for the first time in a year, because, you know, COVID cases are going up. I don't think I need to tell you everything that goes on with long COVID. We're just beginning to understand it. And I attended a Keystone conference earlier this year in New Mexico. And I think that was a good location because long COVID work is the wild, wild west of clinical and translational research. And I will die on that hill because I heard about 50 people give talks and every one of them had their own theory and data to back it up. I mean, you can't all be right, or can they? So we are looking at this and we're taking those mice and we're giving them MA10 and we're carrying them out for months. they get an acute infection, they recover. Tulane has the only currently approved protocol to take mice out of BSL-3 once they've recovered and they don't have virus in their repeat stool samples after about 2 weeks. So it's hard to do cognitive and functional testing in mice under BSL-3 conditions, right? Because it's by very definition, it's very restricted. But once you can get them out of there, you can do a lot. And so we're actively doing that right now. Some of our early days work shows that if you look weeks and months after infection, their brains don't look like someone that's not been infected. And this picture on A, every time I show it, I get asked, is that real? Yeah. And about 30% of the mice, and we don't know why, 30% of the mice that get infected, you look some months later, there's not much going on in the hippocampus by way of neurons. 30%. Now, this is clinically relevant because you may be familiar with the Nature Medicine paper that came out from England, where they had the before and after. So a bunch of, in the UK with their National Health Service, patients were participating in studies that were getting routine brain scans, MRIs, and then the pandemic started. And some clever researchers said, oh, we've got the before, let's bring them back in and look at their brains again. And about 30, 40, I think even higher % of them had hippocampal atrophy and other things associated with, dementia or cognitive decline. These are, healthy individuals. They weren't, already demented. They weren't elderly, 20, 30-year-olds, which is scary. And that's about what we saw. About 30% of the mice, we saw these changes, and I don't know why. Why not all of them? So that's, disconcerting. What is it about those mice? They're all genetically identical, right? They got the same amount of virus. But we saw, increases in microglial activation. This is IBA1, but other markers as well as their morphology. So I want to leave with this. This was a hit when I spoke in New Mexico. This is from a relatively recent paper that says, hey, if you develop long COVID, you've got reduced blood flow to the brain. Simple as that. And that you could take all of your patients, I don't know if you guys have a long COVID clinic, but if you scan them and they've got any neurologic complaints, that's what their scan's going to look like, reduced blood flow. And it's white matter impacted. So I thought, that's interesting, because we have a method of reducing blood flow that primarily impacts white matter, our BCAS model. So we did just that. We took our mice, we put coils on them, we waited a week or two, we infected them with MA10, and it sort of makes everything worse. We see increases in alpha-5, we see a lot more of an increase with COVID. We look at astrocytes, we see astrocyte activation go up. After BCAS, we see a lot more of it with COVID, et cetera, and so forth. And so we got, actually, earlier this year, we got an NIH 5-year grant to look at this. Superimposing, this was before anyone acknowledged that there was an association with any sort of mild cognitive impairment or pre-cognitive decline in COVID. And since then, the NIH has put out calls for proposals to look at the relationship between somebody who might become demented one day, gets COVID, and then 6 months later has clear symptoms. So there's an association. I think it's cerebrovascular in nature. This is more of the same. We do BCAS, we see changes, we add COVID on top of it, it's a lot worse. I like this picture. It's looking at a myelin. Generally speaking, reduced myelin staining in the brain is not good, and it's a lot worse after COVID in our cognitive BCAS model. So I'd love to show this picture because this was during lockdown, and we had our lab meetings remote. And there are 3 people in this picture that have COVID. And if you scan it, you'll probably be able to pick them out. But we kept going through the entire lockdown. And a lot of those studies that I showed you were done during that period. So the work wouldn't be possible without the individuals in that image. And then I'll just post collaborators. Hopefully I did okay for time. Looks like I did. So I'm hoping to get some questions because I threw kind of a lot of ideas out there. But you'll see for me, everything sort of flows. I started thinking about matrix and then matrix receptors and how they impact the blood-brain barrier. And I think everything is sort of stemmed from that. And I bet pretty early on that COVID played a key pathologic role in vascular dysfunction. And I think that's where the field has moved. I think a lot, not all, but a lot of long COVID can be explained by inflammation and vascular barrier disruption. So that's what we're actively looking at now. Thank you very much. So time for questions? Yes. So we haven't done like a cardiac arrest model, if that's what you're getting at. Or have a colleague that does that in rats where you completely cut off the carotid arteries. We just haven't gotten around to looking at that yet. Man. So that's a challenge in all of our stroke models. If your therapy has benefit, everything you look at is going to be better, right? So everything we've looked at in vivo, we've also looked at in vitro. So the models aren't as good to look at barrier integrity, cellular monolayers, et cetera. But we've shown pretty consistently that if you take an endothelial cell monolayer, put it under laminar flow, add astrocytes, you get a certain barrier tightness that's impacted negatively by reducing oxygen and doing other things, and ATN can mitigate that. So the flip side to all of this, a recent paper came out from a group in Oregon looking at alpha-5 beta-1 in ALS. And it turns out that in humans with ALS, as well as the SOD1 mouse model, alpha-5 beta-1 goes up in microglia and endothelial cells in impacted areas of the spinal cord and brain. And they think it has everything to do with the inflammatory cells getting into the brain. And turns out that alpha-5 beta-1 plays a key role in trafficking. So in our stroke paper, we actually looked at this and saw a reduction in infiltrating inflammatory cells, which I posited was secondary to the barrier being tighter. But it's just as likely that it was, they couldn't get in because they couldn't access the integrin. So you're right, there's possibility that there are other things happening. But we started with the barrier because it made sense and I could test it in cells as well as in vivo. Okay. Yay! See, that was my goal. I kept looking. He's awake. Okay. Other questions? Oh, don't be shy. Come on.
So for the ATN or the protein, if you use it like a few hours after the stroke happened, because in the lab, I think you use the stroke and then maybe right away you start the experiment. Did you try to do that like after 5-6 hours after a day, and we still keep eating the same?
Yep. So the study that we published, we waited. I think we gave it right after, and then 1 day, 2 days, 3 days, because we weren't 100% as to when the receptor would be upregulated. So the problem being is there's not a lot of alpha-5 early on after a stroke, but this ATN can also bind to some other integrins that are present. We didn't know whether that was a good or a bad thing. So when you're trying to get some data, you're like, okay, let's make a wide therapeutic window and we can try to focus it later. And so the other thing I almost always get asked about is, well, if angiogenesis is good subacutely and chronically after a stroke, and you're giving a peptide inhibitor that might block angiogenesis, could that be an issue? And the answer is we don't know. So we haven't yet done the studies where we waited days to initiate the treatment. So I don't know whether that would be good or bad. But we do know, so in other words, if we induce stroke, wait 3 or 5 days, then give the therapy, because that period of time is considered a repair and recovery window. If you block angiogenesis with the peptide, would that be deleterious to recovery? So that's really kind of a to be determined question. But like I showed before, if we give it relatively early, within a few days, and we look chronically, we see benefit, which seems to further improve. So that's relatively new. So what I like about it is it's been I didn't mention this, but it's safe, it's well tolerated. These were sick glioblastoma, that's a pretty bad tumor, right? Almost 100% lethal. These patients tolerated it under those circumstances. So I think, we could reverse translate it and then get it actually into clinical trials for stroke and other things.
The hemoconvert, like the bleeding after this does not increase after.
We didn't see bleeding whatsoever. I love when I give grand rounds, I don't miss carrying a pager though. Other questions? No? All right, well, thank you very much for your attention. Really appreciate it. Oh, my pleasure. Could we delete my talk just because there's some unpublished stuff on it?
Oh, I see.
I'm not that paranoid, but
No, I will do that. If it